Hi, and welcome back to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all of those ASPB members who are joining us today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. Today is the 19th talk in our Plantae Presents webinar series. If you haven't already done so, check out our archive to watch all of the previous talks. You can find information on our blog and subscribe to our YouTube channel for more information. If you are having trouble connecting or need to leave the webinar early, just know that a recording of this webinar will also be made available, along with all of the associated materials within a few days. We are really glad to see you here today because we're trying something new. This is our second Plantae Presents panel. Today, and on the dates listed here, we will have shorter 7-minute talks featuring the work of 4 researchers. After their presentations, we will open a panel discussion at the end for you to interact with our speakers. We hope that this will allow us to include even more amazing presenters in this series and make these webinars even more interactive. Visit our website to learn more and apply to be a panelist. As you listen to each talk today, add your questions in the Zoom question and answer Q&A section. You'll notice that there is also a chat area open. This section is for you to introduce yourselves, say where you are from, and connect with your colleagues. Just remember that questions specifically for our panelists should be shared within the Q&A section to make them a little easier for our moderator to navigate. Javier Moreno is with us today to moderate this discussion. Javier is currently a researcher and adjunct professor at the National University of Litoral, maybe pronounced that wrong, in Argentina. He studies plant-herbivore interaction and associated transcription factors, controlling the plant-inducible response in Arabidopsis and Mercantia polymorpha. Javier is also very involved in scientific outreach. He is an associate feature editor at ASPB's journal, Plant Physiology, and he is also part of our Plantae Presents steering committee and the inspiration for this new panel format. I'm going to turn it over to Javier now to moderate today's session.
Hi, Katie. Thanks. Thanks to the speakers for joining in today. I am Javier Moreno, and I am happy to be here to introduce these talks that are about the Evo-devo field. And one of the things we'll figure out today is that it's actually a pretty broad field. And the speakers today will show that basically, that you can have very different stuff doing on this field. I would like to say that in the current COVID context, this format of oral presentations was actually thought as a place to increase the visibility of young plant biologists from different geographical regions. And we actually made a good job. Today we had people from India, from Argentina, Chile, and the USA. So we really think that this format can help young researchers to show their work, and of course getting feedback from the people, from colleagues, and also to foster collaborations in a time that is hard to attend to a meeting or to meet people personally. So hopefully we can help in that direction.
My name is Saavedra, and I'm a researcher from CONICET in Argentina. I would like to share with you the results from our group about the roles of autophagy in the most Physcomitrium patens. Briefly, we found that the protective role of autophagy under nutrient deficiency is conserving B patens, but also provide evidences of this mechanism to sustain the apical growth of protonemata during darkness under optimal growth conditions. So, Autophagy is a highly conserved catabolic process in eukaryotic cells triggered by environmental or internal cues in which cytoplasmic components are sequestered into double membrane structures called autophagosomes and then are delivered to the vacuole for digestion and recycling. Autophagy is virtually involved in all aspects of plant physiology. Most knowledge and major contributions in the field of plant autophagy arise from studies in seed plants. However, in the last years, availability of newly sequenced genomes from algae and bryophytes allow us to dive into evolutionary aspects of plant autophagy as well as species-specific roles of this mechanism. In particular, we are focused on bryophytes and are studying using the most Physcomitrium patens as a model system. P patens is also a model system for apical growth because this filamentous tissue, composed of two different types of cells, chloronema and caulonema, grows by apical extensions. These cells exhibit different characteristics. So while chloronema cells are rich in chloroplast and perform a photosynthetic function and have slow growth rates, caulonemata cells are poor in chloroplast, have a function more related to substrate colonization and nutrient acquisition, and have a faster growth rate. So the questions we want to address are, first, are autophagy, core machinery, and responses to nutrient deficiency conserving P patens? And second, does autophagy have a role in apical extension and or differentially regulate the nutritional state of these cells? So we first searched for autophagy ortholog genes in the P patens genome and found that the core ATG machinery is present. Then we select ATG5 and ATG7 genes and generate local knockout lines and perform nitrogen and carbon starvation treatments considered as canonical methods to induce autophagy in plants. So what we can observe in the figure and in the graph of chlorophyll fluorescence, ATG5 and ATG7 lines were hypersensitive to carbon and nitrogen starvation. So here we have the treatments with light without nitrogen, darkness or darkness and sucrose. These results support a role of autophagy under nutrient deficiency. In addition, we noticed that even the mutants under optimal growth conditions start dying earlier, as you can see here by the yellowish color in the center of the colony. Next, we analyze the protonemata of these mutant cells under optimal growth conditions, and we could observe a high number of dead cells in the 7-day-old tissue in comparison to the wild type, pointing out that autophagy has an important role in moss. Then we measured the lengths of protonema subapical cells, of chloronema cells, and caulonema cells, and found that both types of cells have shorter cell lengths compared to the wild type. In addition, we measured the growth rate of also the growth rate of chloronema and caulonema cells, and whereas in chloronema cells we didn't found significant differences, in caulonema cells we found that ATG7 mutants grow slower than the wild type. These observations lead us to analyze if autophagy was induced in protonemata under these conditions. So we measured the expression of the autophagy marker gene ATG8 after 16 hours of light period, which is our control, and then we compare it with sample of 2, 4, 8, and 25 and 24 hours of darkness or darkness supplemented with sucrose. The 6 pp ATG8 genes were upregulated at night at 2 with a peak at 4 hours and then diminished at 8 hours. or were highly induced in the case of extended darkness with or without sucrose, showing that autophagy is induced in darkness. So we next analyze autophagy induction, monitoring the presence of autophagic vesicles in this tissue using a fluorescent line, which is a GFP ATGB line under the control of its endogenous promoter. We found that in the apical Chloronemata cells, autophagic vesicles accumulate significantly at 8 hours of darkness in comparison to light conditions or darkness of subapical cells. However, in Caulonemata, the accumulation of autophagic vesicles was already at 4 and at 8 hours, indicating an early induction in these cell types and thus a differential response of these cells. To conclude, the data present here show that the protective role of autophagy under extended carbon and nitrogen starvation is conserved in Physcomitrium patens. In addition, our data provides a relationship between autophagy and apical growth, suggesting that autophagy could sustain the growth of protonemata cells, in particular that of Caulonemata, which exhibit a higher growth rate. Finally, I want to introduce our research group led by Professor Ramiro Lascano, and I want to thank these institutions for financial support, and thank you for your attention.
First, I would like to thank Plantae and organizers for giving me this opportunity to present my research. Today, I would like to talk about the morphological diversity and underlying genetics of abscission zones in grasses. Abscission is the process that plant organs detach from the main body. The fruit abscission, also called fruit shattering, is very important for seed dispersal and offspring survival. However, it is an unfavorable trait in agriculture. During the history of domestication, non-shattering or reduced shattering plants were selected repeatedly and independently in different varieties of crops. My research is trying to understand whether the genetic basis of this process is conserved or not. I use the grass family as my research system, which contains the most important staple crops, rice, wheat, and maize, and also many minor and orphan crops and wild grass species that have the potential to diversify our food choices facing the challenge of climate change, the increasing global human population, and the sustainability of the ecosystem. Understanding the mechanism of fruit shattering in different grass species is crucial for better utilizing these natural resources. It is long known that grasses shed their fruits at different positions along the inflorescence, and the abscission position is often used as a taxonomic character. To understand whether the different positions follow a phylogenetic trend, me and my previous undergrad intern Aditi, we mapped these positions of different grass species on a grass phylogenetic tree. Here, different colors represent different abscission positions. You can see that they are generally conserved in closely related clades. We reconstructed the ancestral state of the position, which is shown in blue color, is directly below each floral unit. So we want to ask a simple question, whether the different positions of abscission reflect different developmental process of the abscission layer. With the help of 2 RU interns, Rachel and Patty, over 2 summers, we together selected 17 species and observed their anatomy. Abscission occurs in specialized cell layers called the abscission zone. The anatomy of abscission zone is often distinct compared to the surrounding cells. For example, in rice, which is extensively studied for the genetic basis of abscission zone development, the AZ is composed of smaller and non-lignified cells surrounded by larger and lignified cells. We want to see how much conservation of this pattern is among different grass species. Here shows you some examples. We did identify species with similar anatomy as in rice, such as Chasmanthium. However, this pattern is not always conserved. For example, in Brachypodium, although the cells in the AZ are smaller in size, it is more lignified compared to the cells surrounding it. In Zea maize teosinte, which is the wild ancestor of modern maize, only one side of cells are lignified, but not the other. While in Saavedra viridis or green millet, there's no obvious difference in terms of cell size or lignification pattern. These results suggest that the developmental program of the AZ may not be conserved among the grasses. To further understand the molecular mechanism controlling the AZ development, we selected 3 species that have available reference genomes: weedy rice, Brachypodium dystryon, and Saavedra viridis. Both rice and Brachypodium have the ancestral AZ position, while Saavedra has the derivative AZ position. The AZ of rice and Brachypodium are also smaller in size, while all 3 species have some differences in terms of lignification patterns. I dissected the AZ and tissues immediately above and below the AZ, which I called upper and lower regions U and L. I selected 2 time points before and after amphysis and performed RNA-seq analysis. Using weight gene co-expression network analysis, or WGCNA, we identified modules that are specifically expressed in the AZ either at a younger stage or the older stage. The numbers in the parentheses shows the number of genes in these modules. We compare the overlap of these orthologs between species. To our surprise, the overlap is quite limited. If we look at the overlap in all 3 species, only 2 genes were identified, including MYP26 transcription factor and the lysine decarboxylase family protein. They are both enriched in the AZ at a younger stage. This MYP26 is interesting because their adoptive ortholog also plays a role in anther dehiscence. So we decided to validate the results with in situ hybridization. We were able to detect specific expression in the AZ in both rice and Brachypodium, although the signal cannot be detected in Saavedra. To summarize, loss of fruit abscission is an important domestication trait in cereal crops. The AZ differ in anatomy and cell wall composition in different grass species. Spatial gene expression patterns are divergent in different grass species regardless of the AZ position. MYP26 may play a conserved role in AZ development. With that, I would like to thank my postdoc advisor Toby Kellogg and the other lab members and our collaborators. If you are interested in more details of the work, please contact me or check the 2 publications here. Thank you very much for your attention.
Hello, my name is Francesca Diaz, and today I'm going to talk to you about the present and past relations between plant diversity and climate change in the Atacama Desert. A research that I'm doing as a postdoc in Rodrigo Gutierrez's lab and in collaboration with Claudia Torres' lab, both at the Pontificia Universidad Católica de Chile and with a team from Landcare Research in New Zealand. Understanding long-term ecological dynamics requires detailed reconstructions of the history of past environments. We try to predict what will happen with future climate change, but we still don't understand what happened in the past and how ecosystems and plant communities respond to those changes. So our main objectives are to describe the present and past plant species richness and diversity, and to develop a long-term plant climate model to understand how plants respond to a changing environment. With pronounced rainfall seasonality and intervernal variability, the desert vegetation of the Atacama can be challenging to study regarding short-term ecological responses. We have been monitoring plant species in an elevation transect during the last 10 years in the Atacama Desert, and we have found 70 plant species in the sites. Here I'm showing 8 years of rainfall in black and plant richness in green in these sites. You can see how total richness responds to accumulated rainfall. But well, will happen when we go further back in time. Here, I'm showing the last 40 years of rainfall, and you can also observe that the time window that we have been monitoring may be a good sample of what has been happening in the sites during the last decades. Also, here I'm showing that the decadal magnitude of rainfall variation, here in black, could be related to an ENSO or El Niño index in red. But when I talk about the past in this research, I'm referring to the last 30,000 years. So in this time scale, the magnitude of decadal variation is almost imperceptible, as here. So at this scale, we can observe the last glacial period, or transition. During the last glacial, temperatures here in blue were around 8 degrees lower than today. Here, when the temperature started to increase, the Atacama shows two pluvial events here in gray, when estimated precipitations are more than double than today, and the lake levels and groundwater recharge increase across the desert. So this is the kind of climatic variability observed at the millennial timescale in the Atacama, and we want to understand their impact on vegetation. So coming back to our objectives, after monitoring our elevation transit during the last decade, we have described present biodiversity. But how can we obtain past richness and diversity to develop a long-term plant climate model? And we use exceptional field camp systems. These rodents are the best plant collectors. They live in middens inside caves and collect all plant species around their nest and deposit them inside their middens. So after some years, some of those middens got fossilized, and you can find them in the desert and obtain their formation date using carbon-14. Around 1,500 fossil rodent middens were collected by Claudio Torres' team in northern Chile. This is a unique record from arid ecosystems, and the reconstruction of past environments often relies on the interpretation of plant macrofossil and pollen recovered from them. Yet to advance our understanding of past environments, new methods are necessary. In this way, ancient DNA is an excellent candidate. So we sequence plant ancient DNA from Atacama middens. All middens were previously collected and radiocarbon dated by La Torre and collaborators. To recover the damaged ancient DNA and avoid modern DNA contamination, subsampling, extraction, and amplification were performed at the specialized ancient DNA laboratory at Landscape Research in New Zealand. The subsampling consisted of irradiating the midden sample with UVC, removing the outer surface, and taking a piece for ancient DNA analysis from the interior. The ancient DNA extraction and amplification was made inside the ancient DNA laboratory. The sequencing was made using next-generation sequencing in a modern DNA lab, and finally, the taxonomic identification or data analysis was made using a local taxonomic barcodes database and using GenBank. We obtained a 71% of success in the extraction and amplification of fossil middens, and the oldest plant sequence was of 28,000 years old. We identified 51 plant taxa from our local database and 70 from GenBank, with a total of 82 plant taxa. We use radiocarbon-dated fossil rodent middens and plant ancient DNA to describe plant community changes up to 28,000 years before present. In this figure, species or genera appear here and the time here, from present to the past. So you can see how plant taxa change across time. The most significant changes compared to the present occurred from 17,000 to 7,000 years before present, when plant species were found up to 1,000 meters below their current distribution, as we can observe here in this present-day conditions scheme, and here, how it could be during these pluvial events during the past. Finally, how vegetation adjusts to shifting environmental conditions is relevant for developing more comprehensive diversity studies and models. For example, here, regional climate models for northern Chile predict temperature increases around 4 degrees higher than today, and decreases in precipitation around 10 to 30% lower than today over the next century. Such potential future climate change is similar in magnitude to that which occurred during the last glacial interglacial transition, and over a timescale of several thousands of years. So in a fast-changing world, Atacama plant communities will most likely undergo profound changes. A long-term perspective and integration of paleoecology and conservation biology will need to be adopted if the vibrant Andean Atacama ecosystems are to be conserved for the future. Thank you very much. Goodbye.
Hi everybody, this is Kumud. And this is Aditi. And we are here to present our work on comparative transcriptomics to understand light and temperature signaling in evolutionarily divergent plant species.
Environmental cues such as light, temperature, water, binding factor, they affect virtually every aspect of plant growth and development from germination to senescence. Light and temperature are two such environmental cues that are very well studied in the modern plant species Arabidopsis. The Arabidopsis responds to light or low light or shade condition by showing hypophotic elongation of the petiole, and by showing early flowering. These responses or these morphological responses are collectively termed as shade avoidance response. Similar morphological responses are seen under high ambient temperature conditions, and they are collectively termed as thermomorphogenesis. So our interest was drawn to study the developmental response and the underlying molecular networks or molecular pathways in evolutionarily diverged species. We used three different plant species, that is Arabidopsis thaliana, Solanum lycopersicum, and Oryza sativa, that are different in their plant lineages and also are grown at different geographical locations. We first asked what would be the morphological response of tomato and rice towards shade and high ambient temperature, and what would be the underlying mechanism or the transcriptional response of the three plant species, that is Arabidopsis rice and tomato. And the prime objective was to see the similar or dissimilar molecular signatures of light and temperature signaling in these species. And since these species belong to different plant lineages, we have also got interested in seeing how conserved and diverged would be the pathways in light and temperature signaling in these plants based on the orthology of the genes. This slide here summarizes all our results from the phenotyping. C here represents control condition, SH represents shade, HT is high temperature. Shade was given as simulated shade by using low light conditions of low red to far red of ratio 0.5. High temperature was given by using 5 to 6 degrees Celsius temperature above the ambient temperature range of the respective species. Arabidopsis here shows typical elongation response under both shade and high temperature conditions. Tomato seedling showed elongation response under shade conditions, while there was no significant change in the hypophotic length under high temperature conditions. Rice showed elongation response in the entire seedling height, while there was growth suppression in the high temperature conditions. So it seems that the response of shade is universal, but response to temperature is species specific. To get an insight into the molecular networks or molecular factors that could be regulating this universal shade and the tissue specific high temperature response, we did transcriptomics analysis in the three species.
We obtained reads and performed differential gene expression analysis to obtain DEGs under shade and high ambient temperature with respect to control in all the three species. The figure here summarizes shared and unique number of DEGs in all the three species in a tissue-specific manner. We observe here that the response of high ambient temperature was more pronounced with this as compared to shade. Also, we found that larger number of DEGs were unique to the treatments as compared to the DEGs shared between the two treatments. Further, we performed hierarchical clustering analysis to cluster DEGs based on their gene expression profile, reporting coherent expression pattern. We've obtained some very good clusters for shade and high ambient temperature. Further, to identify the biological processes, gene ontology analysis was performed for these clusters. And we identified processes like hormone stimulus, cell cycle, and leaf development. Since our prime interest was to capture early transcriptional response, we focused specifically in the differentially expressed transcription factor families. BHLS transcription factor family is one of the most important family known to regulate light and temperature signaling in plants. Here we present our results for BHLS transcription factor family in all the 3 species. Now, phytochrome interacting factor is a BHLS transcription factor found at the interface of light and temperature signaling. And we found several PIFs in Arabidopsis, tomato, and rice enriched for shade and temperature conditions, suggesting the evolutionary conserved function in light and temperature mediated seedling development in the 3 species. Now, the final question that we asked is to identify conserved pathways under shade and HT across the 3 species. Gene orthology was determined and analyzed by identification of the orthogroups across the 3 species using a tool called OrthoFinder. Now, the overlapping genes between two set of DEGs was estimated by comparing one set of DEG to other set of DEG and identifying the overlap between them. This way we found that around 30 to 40% DEGs were conserved between Arabidopsis and tomato under shade and temperature, and between Arabidopsis and rice under shade and high temperature. We observed further the conserved processes involved in shade and temperature through GO analysis. And here we can see that hormone response like auxin stimulus was conserved in all the species across all the treatments, that is under shade and temperature both, while basinosteroid stimulus was specific to shade conditions and jasmonic acid stimulus was specific to high temperature conditions. Cell cycle and cell division category was also enriched for all the comparisons made here. Thus, we identify the biological processes with similar gene expression indicating their conservation of the underlying molecular network. In the end, I would like to acknowledge Dr. Ashish Ranjan, our lab members, NIPGR, and funding agencies. Thank you.
Well, thank you very much for the streaming. And while the questions are coming in, there are already some in here. So the first one is actually for Laura. I will ask you to introduce yourself shortly. And the first question is related to how did you pick these 2 candidate genes? No, you have in the process of autophagy in Physcomitrium, you have several ATG genes. How did you pick those? No?
Okay. Well, my name is Laura Saavedra. I'm a researcher from CONICET in Córdoba, Argentina, working on autophagy in bryophytes, specifically Physcomitrium. So regarding the question, in the process of autophagy, so when the cargo is selected for degradation, a structure that is called the protophagosome structure is need to get formed. And there is a gene that is ATG8, which needs to be lipidated with phosphatidylethanolamine to be attached to the membrane, and the process starts. So for this process, ATG5 and ATG7 are involved in this lipidation. So we know if we knock out these 2 genes, for example, we know that the process will be completely stopped. And actually, we have a lot of mutants of different plants of ATG5 and ATC7 to compare with. So that was the double reason that we select these two genes.
Related with that, there is here a question from Georgina Fabro. She's asking, is Physcomitrium have specialized cells or tissues dedicated to perform autophagy, or the ATG genes are expressed and function in both tissues that you were characterizing, no? The chloronema and caulonema.
Wait, I will read it again.
Okay.
Well, actually, we believe that autophagy, I mean, autophagy is homeostasis, is necessary as well as a process for homeostasis. So I believe that all genes for autophagy are expressed in all cells, but in a basal situation. However, what we know from other plants is that they turn it on normally in conditions of stress, biotic and abiotic, and as well in different moments of development. So in what we are seeing actually now is that they are expressed in protonemata, in chloronema and caulonema tissues, and actually in reprogramming. I mean, you can take a leaf of moss and do a gunding, and when you follow after this protonemata will grow, and actually we know that all these genes from autophagy during reprogramming of the new tissue as well are turned on. But we don't know, and then as well, a publication recently showed that autophagy in mosses is important as well for gametes development. So that's what we know so far.
There was another related question that is, do ATG mutants present defects on gametophore development as well, or it's only limited to the growth of?
No. In gamete, a group of Sweden just already published that they are important. They were working actually in the development of the sporophyte, and they found that they were important for gamete, for sperm and ovule development.
Well, thank you, Laura. We will move to Yunqing Yu from St. Louis, Missouri. There are a couple of questions for you as well. Facundo Romani asked how do you think is the ancestral state of grasses abscission zone compared to extant crops? You know, how comparable, how much do they look alike?
Oh, hi everyone. I'm Yunqing. I'm a postdoc with Dr. Toby Kellogg at the Donna Danforth Plant Science Center. For the question of the abscission zones in extant crops, first, most of the modern crops, they have lost their abscission zone because they have been domesticated. But looking at their wild ancestors or wild relatives, a lot of them actually don't have the ancestral state of the abscission zone position. For example, rice has the ancestral state position, but in crops like wheat and barley, which is in a different subfamily, they have a different position. And also for the maize and sorghum, they also have a different abscission zone position. So in fact, a lot of our major crops, they don't have the most common position for the abscission zone.
Yunqing, and when you try to compare the abscission, you are looking at into the fruit abscission, no? So what if you compare the fruit abscission to the abscission of other organs in plants? No? How is that comparison? Are they comparable or not at all?
Because in grasses, they don't really shed their leaves.
Yeah, sure.
So the most obvious abscission zone is the seed or the fruit abscission zone. And maybe another analogy is the anther dehiscence. It has the dehiscence zone, but I don't think there's not really much research on the anther dehiscence zone. But in Arabidopsis, there are people who study the fruit abscission zone and also the floral organ, such as the petal abscission zone. And then based on the genes that have been identified, there are two sets of genes controlling the abscission zones in these two organs. So I think there may be some conserved mechanism, but there's definitely a lot of differences in terms of the genes underlying this process.
And related with those two sets of genes that you mentioned, there was another question. If you, comparing the processes, the abscission process, if you can find especially transcription factors associated with this abscission or not, within grasses or maybe between the two big groups, no, grasses and the other angiosperms.
Yeah, based on our transcriptomic research, we did find a transcription factor, is a MIB transcription factor that seems to have a conserved expression pattern. And the orthologs in Arabidopsis also have a conserved role in anther dehiscence. But we didn't find that many genes or transcription factors. And in fact, we tried to generate mutant of the transcription factors identified in rice. We want to create mutant in other grass species, and we don't always see a shattering phenotype, and suggesting that at least not all of them are conserved. But there is one transcription factor, it's a YABI transcription factor that has been found to play this role in abscission zone in multiple grass species. So I guess the answer is depending on which transcription factor you're looking at. They may have different functions.
So you kind of mentioned that you developed the MIB26 mutant in rice. Is that correct?
For the MIB26, we just started the project and we haven't got the mutant yet. But for the YABI transcription factor, from other groups' research, they found in rice, in sorghum, in Citera italica, they have the same conserved function through genetic studies.
Great. And one last question for you. So what is the association between lignin and the abscission zone? Can you find that association in different plants, or it's only limited to grasses?
Yeah, I think this is our first big surprise, because a lot of the research are done in rice and has such a distinct pattern that the abscission zone is not lignified and the surrounding cells are lignified, and it just creates that inherent weak layer to separate the tissues. But when we look at other grass species, it's not always the case. And in fact, the lignification pattern can be very variable in different species. And then in other species, for example, in Arabidopsis or in tobacco fruit, and they did show that the differential lignification is important for the function, but we just don't know. In fact, based on research, it's not always the case in all species.
Well, thank you very much, Yunqing. Francesca, I have here a couple of questions for you as well. I found the whole story really fascinating. So you show that different plant species, you find mainly changes in the richness of plant species across the time. So one of the questions here is if you have observed any relationship with plant development. So if you look at, let's say, in the root structure and development of those species, if you can associate them to a rainy period or to a very dry period in the Atacama Desert.
Well, thanks. Well, in general, in the Atacama, we found many annual species with tiny and shallow roots, and these are common at the lowest sites, which are close to the absolute desert with almost zero precipitation. And annuals expand and contract their distributional ranges in response to rainfall in the short term. So each year, you can see different annual species after the rainy period. And in fact, during our field trips, it's hard to find sometimes the same species. You need like 6 years to describe the annual diversity in some sites. And when you go up to the Andes, precipitation increases and then appears the life forms, the perennial life forms, mainly shrubs. And in our case, are Parestrefia and Baccharis species. And those shrubs descend to the margins of the absolute desert during the past pluvial events. And so this kind of plants tells us that those precipitation increases were sustained in time, allowing this important ecosystem reorganization because the annual species appear and disappear always. And when you have more rainfall, you find more perennial shrubs and grasses. And when you have almost no rainfall, you found only annual ephemeral after the rains and a few cactus too.
Mm-hmm. So it's actually hard to look at the roots, for example, no? in annual, because that annual species, because it's also on the time of the year you go to the field and look at them. No?
Yeah. So really tiny roots, depending on shallow water and just after the rain.
And there is another question: is if here is there any kind of plant fossil in the Atacama Desert useful to extract ancient DNA? And
like,
maybe the next question is if there is, if it would be possible to go far away in the time, no, farther.
Yeah. Well, from now, this was our first work trying to extract ancient DNA from these plants. So we went to just trying to extract chloroplastidial DNA, and that have many copies and are more conserved. But sure, we can try to recover nuclear DNA. Of course, it's harder, and maybe we couldn't find so much as these chloroplastidial genes. But sure, our idea is to try to recover now more DNA and try to recover ancient genes to understand how these plants in time are adapting to these different changing environments. And going back, we don't know. We are just from now working with these 30,000 species of plants, but in other kind of environments, other people are obtaining DNA from 200,000 years. So it depends on the conservation, and we have to try. But from now we don't have plants as old as 50,000 years from the Atacama. So we could work with that material. But now we try to recover different kind of genes.
But there is another. You are always looking into middens, no? Like rodent. Is there any other kind of fossil that you can use to extract DNA, plant DNA?
Yeah, well, depends on the environment. But in the Atacama, the other kind of sample that we could use it's samples from sediments in lakes. Yeah, so you could extract ancient DNA from sedimentary samples. So then you could extract DNA from plants and bacteria and all the organisms that are living in the lake or around the lake.
Yeah, great. And there is one more question for you here. Is have you identified taxa that became extinct in the Atacama Desert with the last increase in global temperature, or?
No, not yet. And it's really hard to identify an extinct taxa because in the work that I showed you, we were just using one molecular marker or one we just amplify one section of the DNA. So it's really fine to really try to discover an extinct species. You need to sequence more DNA to be sure that you're not looking at a different plant that maybe you don't have in your database, or maybe it was an error in the during the sequencing or yeah. So to really find an extinct species, you have to do a really hard work and probably have some morphological remains that also tells you that probably there are some species that you're not looking today. But it's an interesting but a really hard thing to do.
Well, thank you so much, Francesca.
Thank you.
Well, so now we move to the final seminar from Kumud and Aditi. And there are a couple of questions here as well. So one of the questions from Arpan is if, do you observe the vegetative to flowering transition in a thermomorphogenic view? Because according to the slide you have analyzed, transcriptome from cotyledon and hypocotyl. I'm not sure if I'm reading well if I understand it. Sorry, let's go to another. The apparent lack of response to high temperature of rice regarding hypocotyl elongations is due to differential expression of PIF via epigenetic marks caused by acclimation of this plant to moderate temperatures. Do you find the response similar to the one of arabidopsis of tomato if a higher temperature is applied? That is related actually to the morphological question that I mentioned before.
Okay. Hi everyone, this is Kumud. I'm a postdoc at National Institute of Plant Genome Research in New Delhi, India. And Aditi is a PhD student in the lab of Dr. Ashish Ranjan. So we work in both labs. We both work in the same lab. So I will answer the first question from Arpan. We have focused on the early seedling. So we specifically wanted to capture the early transcriptomic response of cotyledon and hypocotyl because they behave differently in Arabidopsis and as well as in rice and tomato. So we haven't really looked into the floral transition part yet, with just the seedling response. And the other question, yeah.
The other question was maybe asking for something that you didn't show, but this apparent lack of response of to high temperature, of rice. At least you showed that in hypocotyl elongation. If it could be due to differential expression of PIF via epigenetic marks. Have you looked into that or?
Yeah, we first of all, we wouldn't call it apparent lack of response because the response seems to be species specific. So rice is showing a response to high temperature and it is shortening of the seedling instead of like say elongation. It could be that we have used the species specific temperature ranges. Had we used 21 or 28 degrees, maybe rice would show elongation because there are reports in wheat where at lower temperature ranges, it shows elongation. And when moving to higher temperature ranges, it shows suppression. So we are seeing similar results here that we see suppression of seedling length, both lamina and sheath in rice. And we do find the differential expression of pills that are PIF-related genes in rice. And it could be because of the epigenetic changes in them. We are still looking into our data. And our data is not entirely PIF driven because we are also interested in finding new transcription factors in both light and temperature signaling.
So we have found transcriptional signatures that are specific to rice and that are common between Arabidopsis, tomato, and rice. So around 30 to 40% of the genes are conserved, or the genetic responses are conserved in these three species in this early transcriptional window that we have captured in 1 hour. And other DGs or other response seem to be species specific.
And there was another question related to that point, Kumud. If related the species specific genes, can you cluster like dicots? If you look at only in the species specific genes, can you cluster dicots together and monocots on the other side, or not? Those are completely independent of the, well, dependent on the species.
Yeah, that is a possibility. We could separate out monocot and dicot related genes. That is something we haven't reached that far, Aditi. Yeah? Mm-hmm. So we are at the stage of orthology comparison between the three plant species. Then separating out monocots, dicots would be the next step. Mm-hmm. Definitely.
Well, thank you so much for your answers, and I would like to propose you a final question, no? Regarding the Evo-devo field. And maybe my question is, given that this is a pretty diverse panel of people from different regions, geographical regions, how do you feel about what are the biggest or larger constraints or challenges that you find to develop this the Evo-devo field? Can you share your thoughts about it?
Maybe I can start.
Sure, thanks.
I think for me, in my PhD, I actually work on Arabidopsis, and there are a lot of molecular tools, and it's very easy to work on a mutant you just order from TARE. But then since my postdoc, I started to work on different types of grass species. And I guess I'm still lucky that in grasses, there are still a lot of model species or crops that have available genomes and resources. But when you're down to functional studies, when you want to knock out a gene, it's still a challenge, especially if you don't work on a crop species, and it takes time and money. And then in some species, this resource is just not available. I think in that point of view, it's still maybe a challenge, but I also believe that it's a very good time to do Evo-devo studies because there are more and more research and technologies. Yeah.
Great.
Yeah. Perhaps I can continue.
Right. Sure, go.
Actually, I think it's a really good moment now because for Evo-devo studies and comparative genomics approach, until very recently, we have very little amount of models of non-seed plants, for example, or of algae species compared to angiosperms. Like if you see three years ago in the phytosome, 95% of the genomes that were available are all angiosperms. But actually now, I mean, in the last 2 years, there have been the release of, for example, this initiative of 1,000 plants, transcriptomes of 1,000 plants, including algae. And that, for example, brings us a lot of species of mosses, of liverwort, of hornwort, of streptophyte algae that really have moved forward our understanding about the water-to-land transition, for example. I mean, if you go to these signematales algae, which are the sister clade of land plants, you can see that genes that we thought that we were only in land plants, we now know through these studies that they were already present there, like the peel
the PIL gene that encodes for the ABA receptors in land plants that are really important for biotic and abiotic responses, or grass transcription factors that were already there. So I think all this information actually is allowing us, for example, to understand that actually this plant gained these genes before to overcome the challenges of terrestrialization. So I think with all this information, we are in a good time to make new questions to understand evolution, actually.
Great. Thank you, Laura. Francesca, Kumud, or Aditi?
Hi, I'm Aditi, and yes, I agree with Laura that now we are in good times that so many, other than angiosperms also, we have data for and genome sequence for other mosses and live birds. And now that Earth Biogenome Project has been started, so we will hopefully have many more species sequenced. So yeah, but then I again agree with Yunqing that even like Arabidopsis, that data that is curated for Arabidopsis is highly resolved. Like even if you go for gene ontology analysis or any pathway analysis, which is not that much resolved for other grass species. Like if you go even like rice and tomato, they are model species, but still we face some difficulties for high resolution of pathway or this gene ontology enrichment. So yeah, but hopefully we will find new ways and techniques to do so, new bioinformatic tools and highly resolved bioinformatic tools to do the functional annotation. So yeah.
Great. Thank you, Aditi. Francisca, your final remarks.
Okay. Well, I came from the ecology research area, so I'm used to work under natural conditions and with non-model species. But now when I started to work at the Rodrigo Gutierrez lab, when I'm working now, I started to collaborate in these transcriptome Atacama studies, and it's really complicated to separate the most important factors in determining the results that you are observing. So I think that the main constraint is related to the complexity and diversity of environmental factors under each variable you observe, and not only environmental, because you also have a lot of different organisms interacting, like plants with plants and plants with bacteria and more. So I think that is the main constraint related with this kind of work.
Well, thank you so much. I really enjoyed the webinar. Thank you for joining. Katie?
Thank you, Javier, and thank you for our speakers who presented today. This has been really fun, and I'm glad we were able to try out this new format. Thank you all to our attendees as well today. There was a lot of really great questions that came in, so that always makes it really fun too. I put a link into the chat for our upcoming events, including the panels that are in the works. So if you are interested, please submit your research to be on one of these panels. This recording will be posted within a couple of days, and so if you would like to share it with your friends or share details with other people, please do. Yeah, thank you all for joining, and see you next time.
See you.
Thank you.
Bye-bye.
Thank you again.